Now we are ready to discuss what happens during translation. In order to understand translation, which occurs in the cytoplasm of the cell, we have to meet a couple more players of this process. First is tRNA, which stands for transfer RNA. Okay, so structurally, this is a single-stranded piece of RNA polynucleotide, so RNA nucleotides in a long chain. But because certain part of that chain on one side happen to be complementary to certain sections on the opposite side of the same chain, it kind of folds in on itself in certain parts and creates hydrogen bonds. So here, this is a nucleotide that is complementary to this. So maybe this is C and this is G, and this is U and this is A. So they would kind of clump together. tRNAs have this structure all the time, and there are many different types of tRNAs. What the only difference between them is this 3 nucleotide section at this bottom end that's called the anticodon. It varies in what nucleotides are there. So the overall structure of all tRNAs is the same. However, there is variance in the 3 nucleotide section at the bottom. And depending on what the 3 nucleotide section at the bottom is, the amino acid attachment site will be slightly different. Okay, now that is the structure of tRNA. However, let's talk a little bit about the function of tRNA. Although the letter T stands for transfer, I like to think of it as a translator molecule. So imagine you're going on a trip to Romania. Do you know Romanian? I don't know Romanian. If you don't know Romanian, then when you get there, you're looking for a translator because you're going to be going to little villages and castles where people don't speak English or any other language you know. What is it that you would require of your translator? If you are a native or fluent English speaker, then you would probably want the translator to know both languages, English and Romanian, right? That's what would make a functional translator that is useful to you, knows both languages. These molecules, these tRNA molecules, they work very similarly because they are made of nucleotides, so they know the nucleotide language, and they can read specific sections of nucleotides based on the anticodon section, I'll show you how that happens in a minute. Here on the attachment site, what attaches here is actually an amino acid. So depending on what this varying 3 nucleotide section is at the anticodon end, a different amino acid will be sitting on this attachment site. This right here in the other diagram is a different cartoony version of tRNA that has less structural detail, but it's kind of easier to draw for next diagrams that we have. So the whole function of tRNAs is they carry amino acids, and their anticodon loop contains 3 nucleotide section that is complementary to mRNA. Okay, I will show you how that all comes together in just a minute. We're going to look at one more view of a tRNA so you're really familiar with the structure. Okay, here is kind of a flattened version. If you were to take the one that we looked at together already, and you kind of flattened it to be able to see. This is how they call this the cloverleaf model of the tRNA. So you can actually see all the nucleotides that make up the tRNAs and how they're almost all entirely the same. And what matters is the anticodon end that's going to be reading the message on the messenger RNA. The next important player in this action is the ribosome. Okay, let's look what a ribosome looks like. It's going to have 3 specific binding sites for tRNA, and they are called P, A, and E sites. But let's review the overall structure of ribosomes. Remember, ribosomes are made of, this is just a cluster of proteins and RNA that's stuck in there. There is no membrane on ribosomes, so they are not technically membrane-bound organelles. There's no phospholipids whatsoever. So just proteins and RNA pieces stuck together, forming a large subunit, and different proteins are clumped together to make the small subunit. When the ribosome is not working, it is separated into the 2 different subunits. When it comes together, it begins to function to make proteins. Okay, the way that the ribosome works is it holds the messenger RNA in between the small and the large subunit. They sit in there, and inside the large subunit there are 3 little caves, and they're caves where transfer RNAs, the tRNAs that are the translator units I just talked to you about, there are 3 sites where they can sit. And the sites have names E, P, and A. You do not need to worry about the names of the sites. Okay? So the 2 primary function of a ribosome is to decode the mRNA and form peptide bonds. Remember that peptide bonds are covalent bonds that are formed between 2 different amino acids, okay? And there's a special enzyme that is part of the ribosome called peptidyl transferase that is catalyzing the formation of this peptide bond. Now, how does the translation actually take place? We're going to go step by step in just a minute, but one last thing I want to tell you about the so-called dictionary of how you translate words, quote unquote, between the language of nucleotides and language of amino acids. So to go from mRNA to a protein. The words, quote unquote, necessary are made up of codons. So a codon is a block of 3 DNA nucleotides that correspond to a specific amino acid. So what that means is, okay, we're looking at a strand of mRNA. Remember, that's a polynucleotide. So we would have A, U, G, G, C, C, G, C, A, U, okay, for example, right? The way that the ribosome and transfer RNAs are able to read it is they read 3 of those nucleotides at a time. So if there's CCG, right, in a threesome next to each other, they will be read as one, they have one meaning, like as if it's one word. However, how do you know where you start the reading? You do not begin at the very, very beginning and just take it every 3 nucleotides from there. You start only at the first AUG. So every messenger RNA has to have AUG in that exact order, those 3 nucleotides in that exact order, somewhere toward the beginning. And only then do we begin reading. AUG is always the first codon, So the first triplet that is read. And when there's an AUG, it brings, the transfer RNA brings an amino acid called methionine. So the first amino acid that is added to any polypeptide chain or any future protein is methionine, because AUG is the start codon. And that's where you determine how to form your codons, or the threesomes. After the AUG, the next three nucleotides are going to be read together. So they could be ACA or CCC or any of these, until you get to any one of these three stop codons. Then the whole reading and translating process stops and all the players go their separate ways. Okay, so for translation, the complex includes the first transfer RNA that's always going to be loaded. Remember, at its back end, it's going to be carrying an amino acid called methionine, while its anticodon is going to be complementary to AUG, because AUG is always the first codon on the mRNA that is read. Small ribosomal subunit is going to come in first. The messenger RNA that we're trying to translate is going to be present. And finally, the large subunit is going to be added to begin the whole process. Okay, let's see where this is all happening. Okay, so here you have your small subunit of the ribosome. Next thing, you have the messenger RNA that you transcribed in the nucleus and are now ready to translate in the cytoplasm is going to come and land itself on the small subunit. Okay, here in the bottom, you can see how it's already sitting on that small subunit. The first transfer RNA, remember there are many different kinds of tRNAs, and they only differ basically in this bottom three nucleotides. Well, the tRNA that happens to have complementary to AUG, which is UAC, is going to come and land on that AUG, and in its back end, it has the first amino acid, which is called methionine. Okay, so notice how we do not start translating the first nucleotides of the mRNA here. We wait, or more accurately to say, the ribosome and the tRNA, they search for anywhere where there's like, oh, where's there A? Oh, there's A, but the next one is C. Okay, that's not going to work. AUG, bingo. It's going to start at the AUG. The first transfer RNA is going to come, land on that first codon. So this AUG, this three nucleotide sequence, is called a codon. Codons are always three nucleotide sequences. And what is zooming in on is that the bottom part, or the anticodon part of the transfer RNA, has UAC on it. So it will become complementary, can bind temporarily and bring in a methionine. When that is all set up, the large subunit of the ribosome comes and closes on top. You see how there are three sites available, and we have a transfer RNA in the middle site. And that is the end of the setup, or the end of the initiation process of translation. Elongation comes next. During the process of elongation, amino acids are added in order depending on what the next codon codes for. So a second charged tRNA will come, bind in the empty A site, and a peptide bond, which is the covalent bond between two amino acids, is formed by the enzyme peptidyl transferase, which is an enzyme that's a part of the body of the ribosome. And then addition of successive amino acids occurs in the same way. So here, new amino acid has come. It is attached to a tRNA. What determines which tRNA comes here and which amino acid it's carrying? Well, it depends on the triplet or the codon in the next section. The tRNA that comes is the one that happens to have a complementary anticodon, and then based on that, it brings an amino acid. So how do we know what amino acid is going to be there? It's back to reading this. So if the first codon, the initiation or start codon, was AUG, and it brought M, more methionine, as the amino acid, then if the next triplet is CCG, and this is the next triplet on the messenger RNA, then the tRNA that comes would be complementary to this, and it would bring proline. As you can see, there are several different codons that code for proline and most of the amino acids except for methionine and tryptophan. What happens next is we are over here, then the whole ribosome moves as if it's like moving down the messenger RNA in such a way that both tRNAs that were there move to the other positions and A is empty again. New transfer RNA comes depending on the codon here, and it brings the appropriate amino acid. When it binds here, the enzyme peptidyl transferase creates a peptide bond, and the whole line of amino acid gets moved over to the next tRNA, and the cycle continues until all the amino acids that have grown are on one tRNA because they get passed on, and the empty transfer RNAs leave and they get recharged with the appropriate amino acid depending on their anticodon here. They get recharged in a separate area of the cell by different enzymes. And where do the amino acids that get put on these transfer RNAs, where do they come from? They come from the food that you eat. When you eat proteins, they are made up of amino acids. In digestion, the amino acids are separated from one another, and you end up with amino acids in your body that get moved to wherever they're necessary. Some amino acids out of the 20 in our body can be synthesized from precursor molecule or from different amino acids, but some cannot be synthesized at all, and they need to be eaten in their final form from the food we eat. We call those the essential amino acids. Termination is when the ribosome hits a stop codon. There are 4 stop codons: UAA, UAG, and UGA. You do not need to remember any of these. You do not need to memorize any of the codons, but you should be familiar with reading this table. So at any point when the ribosome snakes along the messenger RNA and the next triplet happens to be one of these, it doesn't matter how much more of the messenger RNA is left untranslated. It doesn't matter at all. If one of these codons is hit, then termination occurs. So after the stop codon is recognized, there are some release factors and some enzymes and helpers in the ribosome that basically disassemble the whole unit. The large and the small subunit of the ribosomes separate, messenger RNA is released, and whatever is created of the amino acid chain or the peptide, polypeptide, that gets released as well. So stop codons, there are 3 codons used for termination. In general, the genetic code is the same in all cells of all organisms. And it is degenerate, which means that some amino acids are specified by more than one codon. As I said, all amino acids except for methionine and tryptophan are specified by more than one codon. So the code does not need to be 100% exact. In some cases, you can be lucky, and even if there is a mutation, it will still code for the amino acid that's necessary because of the redundancy in the code. So here it shows that a stop codon was hit. There's a release factor instead of a tRNA, instead of a transfer RNA. Everything dissociates and moves along. The messenger RNA would just get recycled, it would get broken apart, and the pieces would be reused, while the new polypeptide chain would immediately fold in on itself. If you remember that video, The Inner Life of the Cell, which I showed you in chapter 4, when the polypeptide chain is built, it immediately folds in on itself to create its 3D structure, depending on the hydrophilic, hydrophobic forces between the amino acids, as well as negative and positive amino acids that can interact in different ways. As I mentioned, the code is practically universal with tiny exceptions in some bacteria. And this is the strongest evidence among a lot of good evidence that all living things share common ancestry. And this allows us to have some advances in genetic engineering. I've mentioned previously that pieces of the genetic code, so a gene or several genes, can be inserted from one organism inserted into another organism, and they will be read by that organism's machinery and expressed to produce the proteins. So it doesn't matter where that DNA comes from. A yeast cell that has human DNA can read it and produce human insulin, which can then be cleaned up, harvested, put into a buffer, and used by people who can't produce their own insulin. This is true with many other proteins like human growth hormone and others that are used in different therapies. Mitochondria and chloroplasts have some differences in stop signals, so their stop codon is a little bit different. Because remember, both mitochondria and chloroplasts, they have not only their own circular chromosome, their own DNA, they also have their own enzymes within those organelles to read and produce products of those genes. So in review, RNA polymerase creates the mRNA primary transcript, which is a complementary copy of the gene. The gene is on the chromosomes. It's in the double helix, one of the strands of the DNA polymer codes for a particular product, like a protein. A whole set of machinery copies it. The main copying is done by RNA polymerase, more specifically RNA polymerase 2. I'm not gonna ask you about that specifically, but you should know that it is RNA polymerase that makes the messenger RNA, the copy. Then this primary transcript, this copy in the form of messenger RNA, gets edited by addition of the G cap, the poly A tail, and the excision of intron and splicing back together the exons. The exons are regions that are going to be expressed. This is all done in the nucleus. Then the messenger RNA that is mature, the mature messenger RNA, comes out of the nuclear pores into the cytoplasm, where it sets on the small subunit of a ribosome. The primary transfer RNA carrying methionine, the first amino acid, is going to come. Then the large subunit will close on top, and that will begin the translation process in the cytoplasm. After reading the first codon, which will be AUG, all next triplets or all the following codons will be read, and the appropriate tRNAs are going to bring their appropriate amino acids. So all depends on the code of the mRNA. That polypeptide chain, so the chain of amino acids, will be growing until a stop codon is hit, and the whole system disassembles. The amino acid chain, which we call polypeptide chain, folds in on itself, and it can continue to have other modifications done to it. It can join up with a different polypeptide chain to work together, which is the case of quaternary structure for proteins. This is true for some, but not all proteins. And then it'll be ready to do its job. Now, what happens if there is an alteration, a change in the original genetic code, in the DNA code itself? These are called mutations. Mutations are altered sequences of the DNA. They can be divided into several categories, and if there's only a single alteration on that we're talking about, one DNA nucleotide is changed, that's called a point mutation. And they can come in several varieties. For example, a base substitution, it means that at the point mutation, you have a substitution. So instead of one DNA nucleotide, there's a different DNA nucleotide. And this can have 3 different consequences. The consequences can be a silent mutation. It means that just so happens that the new DNA nucleotide still results in the same amino acid being inserted into the growing polypeptide chain. So really, you would never even know that there's this mutation because there is no consequence whatsoever. Missense mutation is when there is a different amino acid inserted because the codon codes for a different amino acid due to this mutation. Right? And there can be nonsense mutations, which are often the worst ones because that is when instead of amino acid being inserted, there is a stop codon instead. So whatever the point mutation was, base substitution, the base that substitutes makes the codon into a stop codon instead of coding for an amino acid. And so if this happens early on in the gene, then instead of having, let's say, 1,000 amino acids in the chain, you can have some small amount like 20 or even 100, which of course would not create a functional protein. So let's look at an example. Okay, so the top 2 strands are the double strand of DNA. Okay, complementary. You see how one is from 5' to 3', the other one's from 3' to 5'. Then when mRNA is made during transcription in the nucleus, then this one strand called the template strand is going to be used as a template to create complementary messenger RNA. Okay, so if here there was a GC pair in the original DNA double helix, then of course you have a C here for the messenger RNA. And if you are looking in your textbook right now at the genetic code, that table with all the codons and what amino acids they make, you can see there's AUG for methionine to start. The next triplet is CCC, and that calls for proline. There's UAU, threonine, CGC, arginine, and UGA stop. Okay. Now, if there was a different change, okay, let's say here now you have a TA, then instead of how in previous you had threonine arginine, you now have threonine histamine. And that is a different amino acid that will behave very differently in the structure and could alter the function of this protein. Okay. In the case of a nonsense mutation, you see that because this pair in the original DNA double helix of DNA nucleotides is such, now it's a TA here, it after proline you have UAA, which is a stop codon, and so it stops and no more of the amino acids are inserted. So this is just like a tiny example. In reality, of course, these are very, very long and have a lot of amino acids. Let's look at a real case example in humans. In order to code for a normal hemoglobin subunit, okay, so hemoglobins are made of two identical alpha subunits and two beta subunits. So you have to synthesize two of the same thing to put together and two of another same thing to put together. So in this particular polypeptide chain, beta, you have this sequence of nucleotides in the DNA. In this particular diagram, they've skipped the mRNA step, so the transcription step. But what they're showing you is if in the DNA you have a T here instead of an A, you will eventually at the end, when amino acids are being inserted, have valine instead of glutamine. Glutamine is polar, valine is non-polar, so it's hydrophobic and it hides inside into the interior of the blob that's supposed to be made. And so you have this angled sticky, I should show you, these sticky ends instead of a nice rounded section that's supposed to be interacting with water that's in our blood. And then these get hooked on other copies of the same hemoglobin, creating these long clumps. And it's like a long stick that's being put into this flexible cell, and it makes the cell sickled. And this one difference in the nucleotide sequence in the DNA causes sickle cell disease, which is a difficult condition for people to live in and requires constant therapies and check-ins with doctors, etc. And up until recently allowed people to only live few short years. Now people are able to live into their 20s or 30s with certain therapies, maybe even longer. Okay, so that's it for point mutations where you only have one, I lied, base substitution mutations where one nucleotide is there instead of a different one. Like there's a T here instead of an A, that's a that's a base substitution mutation. Now let's look at frame shift mutations. This is when instead of having one nucleotide be changed for a different one, you have additional nucleotides there or some that are deleted. Okay, so addition or deletion, usually of a single base, but it could be more. It has much more profound consequences because it changes the whole reading frame downstream of that mutation. So if you take out one nucleotide or insert one, then from there on out, all of the triplets, right, all the codons are going to be altered because the reading frame changes. This can also lead to triplet repeat expansion mutation, where in places where there are triplet repeats, so like you have CGA, 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 for example, it will tend to repeat itself even more after a mutation. And when there are too many of these repeats, it can cause very severe consequences, as can be seen in Huntington disease, which usually shows up in middle age and causes incredible deterioration of brain function. And it's a pretty complex situation here. I'm not going to ask you specifics about triplet repeats, but you do need to know what a frame shift mutation is and what its consequences are. If you want to practice this, then make up a DNA sequence, then on paper transcribe it to make it into a messenger RNA, and then translate it, and then go ahead and insert a nucleotide or delete a nucleotide somewhere in the middle of that original DNA sequence, and then see what the consequences are when you transcribe and translate that new mutated version. You'll see that just there are massive changes to which amino acids are inserted. Okay, that's it for point mutations. And the last bit here is that there can be large structural changes to chromosomes. So changes to the structure of chromosomes can have profound effects. And we've already talked to the fact that having extra chromosomes or missing chromosomes is usually not tolerated, meaning people cannot survive and fetus cannot develop, except for in some rare cases. Well, if big parts of chromosomes are deleted, which means thousands or even millions of nucleotides can be deleted, or when part of a chromosome is duplicated, so a part of a chromosome is copied twice, and then both of those copies are kept in the same. It's like you were copying during DNA synthesis or DNA replication. You were copying, it's like you were copying 3 pages of a book, and then you copied the 3 pages again. So you now have 3 more pages in your book, and there's a section that's repeated. It seems harmless to just have extra DNA, like, oh, it's not, like nothing's missing. We don't have genes missing. So duplication should have smaller consequence, but that's actually not true. And having too much DNA causes surprisingly large consequences to the organism. Can also have chromosomal inversions, which means a part of a chromosome is turned around and inserted in a reversed order. And this could have various consequences. And finally, there can be chromosome translocation, which is when a part of a chromosome is moved to a different location. So let's see here. If you have, this is the chromosome you start with, and we just divide it into several equal sections so we can talk about them. Let's say this section from B to D is deleted, and now the chromosome is missing this major chunk. Of course, as you would guess, all the genes that were on this part of the chromosome, on the DNA double helix here, they're all gone. This can happen when something goes wrong with the spindle apparatus during mitosis or meiosis, or when there is something wrong with the editing mechanism during DNA replication that's supposed to be fixing mistakes of that process. This, depending on what section is deleted and what genes were there, could have various consequences, from some mild health issues to death of the organism or tissue or the cell. In duplication is when this section BCD is duplicated, and now you have BCD, double the amount of DNA of the section, your chromosome much larger. Again, depending on if this happens in meiosis or mitosis, what tissue this is in. Of course, if this happens in eggs or sperm, it has massive consequence, and it's very likely that the zygote will not develop properly. If this is a cell that's like your skin cell, then the cell is likely to just die and will have to be remade. And finally, here you can have an inversion where this section breaks off and is reinserted in the right place but backwards. And this depends on, are you breaking in the middle of a gene here or here? If you're not, and you're just breaking in some filler DNA areas, then this could not actually have any consequence whatsoever. So it varies quite a bit. And then you can have translocations where a section will be moved in completely out of order here. Okay. All these things can happen, and they do happen for various reasons with various consequences. Mutations, however, we're talking here as if they are just really bad and causing lots of health issues. Most mutations are not of any consequence to the organism. They happen in areas that do not code for products, so they do not happen in genes, as most of our DNA is actually not genes, but filler DNA that we don't know what it does very well, or it's regions that control how genes are read. If there are mutations in some of these regions, they have no consequence that we can notice. Next most common consequence of mutations in general is that they are and have negative, harmful consequence to the individual, and they may have disease or death because of those alterations. However, sometimes mutations are good, and they allow for an organism to do something in a better way depending on its environment than it was able to do before. So mutations are the starting point for evolution because, as we learned in chapter 1, in order for natural selection to occur, there must be genetic diversity in a population. How does this genetic diversity happen? Well, one of the ways is mutations. If you're asexually reproducing organism like single-celled bacteria, archaea, or single-cell protists, mutations are your only way to have diversity in your genes. For animals, plants, and fungi, many other organisms who reproduce sexually, we get variety mix from the mixing of our genes with partners, sexual partners. However, in general, balance must exist between the amount of new variation and health of species. And I think that's a good place to end. Balance in life, what a good lesson for us all. This is the end of all of our new material for this term, and I'll see you in class.